Just to sort of introduce the background for this presentation, when we started this ion channel modulator symposium in 16, we promised ourselves that it should not be a product event at all. But actually in 2018 and 19, the audience started asking us, why don't you tell a little bit about your products? We want to hear it. So we decided to, well, apart from having the exhibition that we have had all the years, we decided to, well, we will talk about the products then. But please let us know if it's a waste of time and if you would rather go straight to the drinks or have another more scientific, well, in 25 minutes or so. It's of course a tough competition with the drinks in the Scholar Garden and this weather. But I'll dare. And just like Professor Tucker mentioned, we cannot be here without mentioning our founding fathers, Hodgkin and Huxley, and the work that they did in the 40s and 50s that laid the ground for everything that we are doing nowadays, more or less. They had some pretty serious grounding wire when they impaled the squid giant axon with a silver rod. We have some issues nowadays that makes this more difficult, because later on, Nea and Sackmann were also awarded the Nobel Prize for the development and invention of the patch clamp technique that we are using today. Later on, it became something that we did in many places. This is actually my former boss at the biopharmaceutical company in neurosurge. And again, Professor Tucker actually used some neurosurge compounds in his work. That was nice to see. Where this setup was daily life and is daily life for many of you. In 2001, the normal approach of bringing the electrode to the cell was flipped upside down, and we managed to bring the cell to an aperture in a planar surface, hence also the term planar patch clamping. And then we spent some more years before we came out with the first instrument, Cubet, and in 2005, later on in 13 Cube, and later again, latest and the smallest so far this year, Cubet Compact, and that's what I'll tell you a little bit about. The reason for developing these instruments is, well, multifold. One is that there is a large need for ion channel work. Roughly, channelopathies make up a third of the therapeutic targets of all diseases. And besides that as a driver, there is of course also the scientific, the basic research that's going on in many places. We have done quite a few inventions ourselves, we believe. None will be awarded the Nobel Prize, but still, we actually took an offset with a totally normal manual rig when we did the first approach. We motorized everything that you have to move and find and do and measure. We took a manual rig, motorized the pipette. We made a program that could identify the tip of the pipette, identify the cell, identify the roundness of the cell, and pick the most delicious of those cells that it could see, and bring the two together and measure the resistance, and from there on you do the recording of the resistance and you add compounds and blah, blah, blah. It could also change the pipettes itself, actually. But it was serial in its nature because of one cell after the other. So after the planar patch clamp, where we managed to seal on a planar surface, we came out with Cubet 16, as I said. Later on, it was growing to a 48. Back then we called that high throughput because it was back then. It's no longer considered high throughput. We also invented current clamp because there's a need for that. Current clamp in automatic patch clamp. We also have current clamp in 384 format, and lately, and we have adaptive protocol, something that I'll come back to, where we can interrogate the cell on an individual basis and use its answer to apply to the same cell. And then this year, creating a small instrument that you actually dare share, as we say, because normally I guess not many of you would like to share your manual rig once you have grounded it perfectly well and your micro-manipulator is no longer drifting. You don't want anybody else to tamper with that rig. So this is what it looks like with a silicon surface. You bring the cell onto that, you have an electrode on either side, you have solutions on either side, and we then embed that in a construction that is pretty much the foundation of all our instruments. There are slight differences between the Q patch and the Cube plates, but in principle they are the same, namely some aperture down here, liquids on either side, some pressure to move around the cells and liquid and do the sealing and the rupture of the membrane. And then we multiply that, so either 1648 or 384 at the same time. Yeah, 2005, it got upgraded in 2018, but apart from that, it's, I wouldn't say the same, because a lot of development has gone on during those years. But we were lucky with our timing, or we were good with our timing, because in October 2005, FDA came out with new guidelines for cardiac liability requirements. HERC was found to be very important in in cardiac liability when you do drug discovery and clinical trials. So therefore, there was at the same time a big need for testing HERC liability, HERC blockade, when you were in the clinic with new drugs. So it was, of course, important to show that we can record HERC current and that we can add some compounds and test the pharmacology. So these are the signature traces, you know, with the activation part, which quickly goes to the inactivated state. And when you then step back, you have this huge tail current giving rise to this signature HERC current. We can add compound, and in an IT plot you can see how it is blocked. We of course need to compare to convince the public that it's worth using such an instrument. So comparing manual patch clamp data with QPatch, this is QPatch 2 data and also the first QPatch, and here the ratio between QPatch 2 and manual, just to set to be able to claim that yes, it's coming up with the same results as manual patch clamp, which is and especially back then was, of course, still the golden standard. It's also important to do some current clamps, so we developed that. You have probably heard that many of these planar patch or automatic patch clamp platforms, they take advantage of fluoride. Originally, well, at least when I was a patch clamper, fluoride was actually an ATPase inhibitor to prevent rundown. But it turns out that it actually also glues the cell onto the glass orifice or glass surface pretty well, so it enhances the seal. However, it does also form a brick with divalent cations, so sometimes you cannot have it there. On the Q patches, you don't need it at all because it is glass surface. On the Cube, you benefit from having a little bit of fluoride. So here you can see some current clamp traces where the fluoride seem to have some effect on these action potentials from Core4U cells. So it's good to know that even if it's an automatic platform, you can do it in physiological solutions. To show you a little bit about, well, what do you gain from such an instrument in terms of usability and throughput, I will go through some of the software that you use to command the instrument and give you a little bit of idea of what you can do. For instance, temperature control. You can ask the instrument to run at a certain temperature. The most important thing about that is, of course, to keep a steady temperature no matter whether it's summer or winter, whether your lab is climatized or not. But you can, of course, also use it to test compounds. Not very often compounds are tested at physiological temperatures, but at room temperature. But here you can raise the temperature, and you can actually also lower it as low as you like until the level of condensation on the electrodes and stuff like that. It's good to know that you have a so-called Q stirrer. Everything Sophion is Q, right? Q patch, cube, etc. So there's, of course, a Q stirrer. That's where you harbor the cells on the platform. And you tell the Q stirrer what density your cell harvest is and what density you want it to be when you put it on the plate, and the machine does the rest for you. yeah, that's given again over here. If you do not want to use the Q stirrer, if you have some precious cells that you don't have many of, if it's not a cell line, but if it's primary cells or something like that, you might want to opt to run without the Q stirrer. So you can go as low as totally bypassing it and adding the cells manually and go down to as little as 50 microliters in total for the entire plate. So that's quite a neat feature, even if it's a big machine. Also, in terms of usage of the plate itself, we have an intelligent scheduler, so the Q-pads operate with its pipetting events independently and with feedback from the results. So depending on the criteria that you set up, I will only carry on with my experiment when I reach a certain seal size, if my series resistance is good enough, otherwise try to rescue it. And that's done individually on each site. You can also tell the instrument to run. Here are all 6 columns in a 48 instrument activated, but you can ask it to run only a sub portion of that. And you can ask it to run a sub portion of that and then ask it to roll through the plate. You don't need to be there. So once it's done with, say, the first 2 ones, it will carry on with the next 2 ones. There's some good benefits from doing that. So there was an increasing sort of requirement for throughput. So after having had Q-pads for many years, we decided to go 384. Maintaining is it called not CBS, but SBS standard format. We had to squeeze down all the amplifiers and the plate technology on the same space, and we did so. So there are actually 384 amplifiers embedded underneath the Q chip in that instrument. The ambition with this was to conquer some of the drug screening arena, which traditionally has been done with fluorescence-based technologies. And most people agree that if you have an ion channel, it's better to use electrophysiology than to use a more indirect measure. But you're probably familiar also with the flipper instrument, a very capable instrument that can screen a lot of compounds in a very short time. So we thought that, well, when you do the flipper screen, you're always doing a secondary screen. What if we ask the ion channels the right questions up front, having real gigaseal-based electrophysiology to begin with? Then you can totally omit this secondary screening part and then sort of justify, admit it, the higher price per data point to begin with, because you get better hits, you get better feedback to your medicinal chemist, and can carry on with more meaningful chemistry downstream of this. So that's the idea. So one thing that you can take advantage of when you have electrophysiology sort of upfront and in big scale is, as I said, the individual interrogation of each cell. So imagine the biological variation in, for instance, steady-state inactivation for sodium channels, for instance. If you do a recording, a Boltzmann fit, and say, okay, at which potential do I have 20% inactivation? You will have a spread something like this. 40% inactivation, you will have a spread like that. Spreading the potential across the S-shape of the Boltzmann fit. If you do that on the fly, ask the instrument to then apply the result and run a subsequent protocol in the same experiment, you can see how much tighter the data gets. So when you actually ask for 20% inhibition, i.e. 80% availability of the current, you do get pretty much 80% from all of your cells. The way it's done is that you do a steady state inactivation, you analyze it here on the fly, Boltzmann fit to the result, and then you have at the same time made the next protocol in which you say, in this segment, I want to use the result from that in here. If it's within certain boundaries, and if it for whatever reason falls outside the boundaries, I want to apply a rescue potential, so I still get a result from the particular cell. Throughput comes with success rate. Actually, the flipper only talks about failure rate because, again, admitted, it does get pretty much 100% success every time because it's not relying on a single cell at a time. It's relying on thousands of cells per well. So we are up against a cell that can fail. You don't get a good seal. We have more consumables. We have consumables with 1 hole per side, and we have consumables with 10 holes per side. And we can also customize that if that makes sense. But so we talk about success rates. So therefore, it's good to see that we can actually achieve pretty high success rates. And we can achieve them over a course of an experiment where, in this case, 11 plates have been digested by the instrument. Together with another, specific interrogation of the ion channel in question here, looking for a subtle effect on the inactivation part, which is difficult to find with more indirect measures. And creating what is also important if you are a screener, the so-called Z prime value. It has to be more than 0.5 to give you the screening window that will make sure that a hit is a hit and not just a random event. Having the capacity, the 384, means that you can also embark on cells or assays that are, should we call them, difficult, where if you use primary cells, or in this case iPSC-derived cells, you might not get up to the 90 plus success rates. But if you have 384 attempts in one go, well, 60% is also a lot of experiments. And in this case, we worked a lot with actually the cell culturing conditions, because that turned out to be the determining factor for the success rate, that we cultured them the right way and harvested the right way. And then again, having this massive capacity, you can run with a special delivery plate. You can run control cells together with disease cells, together with disease cells that have been treated in the same run, having exact same conditions on the same plate. So here, as a control measure of sodium current density, and then in a diseased, I should say, the spinal muscular atrophy patients that these iPSC cells were derived from, we can see an increase in the sodium channel current that was then rescued by the treatment with this compound SMN-C3. Comparing with literature data, it looks pretty nice. There was no change in the Boltzmann fit between the three population: control, disease, and rescued. So that's another application of it, apart from drug discovery. It's not only voltage gated that's of relevance, of course, it's also ligand gated. In this case, it's no problem if the ligand gated target in question is not a fast one, so it can sit with the agonist around it for a long time, but some targets are not like that, and they will desensitize and only give you, one shot. So if that's the case, it's important that you can expose the target to the ligand for a very short time, and we stack then the liquids. So the agonist will be the yellow, the wash will be the blue, and yes, there will be a little bit of mixing in the pipette, but that doesn't really matter because we have the embedded flow channels in the consumable, meaning that the mixing zone will be sort of shot past the cell in a fraction of a second. So we can go down to as little as 0.8 seconds exposure of the agonist and get a scenario where you can stimulate over and over again. That's actually customizable, this time delay, whether you want it short, medium, or longer. We can also apply, we heard about applying a compound from the inside. That's also possible to do that on the fly where it exchanges the intercellular solution on the fly. So in this case, there have been shown internal application of 9AC on CLC1 channel, just to show that that's also possible. All right, there are some words about Cube 384, and then as our newest, which is Cube Batch Compact, as we call it. What you cannot really see on the picture here, but what you can see in real life in the exhibition, is that it has 8 holes. You can see them up here. And you can execute your experiments totally independent on each of those. So you don't have to run a full plate at a time. You can run one experiment at a time. You can remake your mind during the run, and you can change the protocol just like you would do on a normal, not on a normal, but on a manual rig. The only thing that you have to do the same is the voltage protocol, so the way that the cells are being sort of captured and sealed and and turned into a whole cell. You set up your compound application scheme. It's in order to sort of avoid the need for having a notebook. You can write down in the program on the screen. It's on the touch screen. This will help you if you say, I want to make a 6-point dose response. You just enter the highest concentration and the number of concentrations and dilution factor, and it will write this for you and keep track of it for you, even if you actually do the pipetting yourself. So you do manual pipetting, and there's a countdown to help you. You click on a button, it will count down for you and say now, and then it will put a line here saying this is where I set now, and provided that you do what you're told, then this will actually add up. So you can see here a scenario where you have again a voltage gated sodium channel that has been exposed to increasing concentration of tetracaine with an online IT plot. So you have the feedback or the readout online in front of you based on where you have set your online cursor, just like you know from, well, HECA or whatever. And you can have various parameters read out at any time from any of the 1 through 8 sites. You don't have to, as I said, pick all of them. In this case, we are running all 8 at the same time. There is a finite waste volume in the machine. The same goes with Q-pads. That keeps, of course, a liquid accounting automatically. Here, you will have to obey, again, the liquid amount that you add, and then look here, have I now filled up the waste? There's room for 250 microliters, and you only add 5 microliters at a time when it comes to the compound, so there's plenty of room for many, many additions. Then you do the analysis, and you will end up with some nice curves. And speaking of analysis, we don't only make the hardware and the control software for commanding them, we also make the software to do the analysis. And we have actually, the latest years, decided to make Sophion all you need, also in terms of the analysis. So we have expanded the analysis package so you can do, well, anything you would normally do. You can also make it present the curves in a nice manner. And if it's not good enough, or if you have some wishes, if you come across our platforms, we are more than happy to receive wishes. But in principle, at least, anywhere from the raw trace acquisition, of course, ITIV plots, concentration response, IC50, V50s, or whatever. And then when you have digested some of these data, you can actually use the resource and feed them back until the raw data level in a circular manner, so you can normalize against one of your digested results, if you like. Having this sort of massive amount of data, it's important that you don't have to go through manually all of your experiments. So quality filters where you set up, I will only accept my experiment if it has such and so high seal, or series resistance, or whatever. That is also possible. So you don't, otherwise you will end up drowning in the amount of data. And once you've set up your project analysis, the next plate you run will be analyzed the same way. So you make that into sort of your template. If you like, you can of course get the data out of the instruments or the databases and handle them by third-party software. There's no problem in that. And then we want you to take care that you don't lose your data, so there's an automatic backup functionality that will run at your convenience. So to sort of sum up and and align them, not necessarily comparing them, but still. Our little new one here, you can run 1 or through 8 fully independent experiments. In the Q-patch, 6 columns with 8 each. You can decide to run n numbers of those 8. And in the big one, you run all 384 at a time. We do whole cell and perforated patch, by far mostly the whole cell. You have to do the pipetting manually on the little one, whereas it's automatic down here. I'll not go through all the details in this scheme, of course, but just remind you that, of course, you can do the sampling at a high frequency, and you can filter it. And also all these, features that you would expect from genuine high-fidelity electrophysiology is available on these platforms as well. So with that, thank you for your attention.
Thank you. Does anyone have a question? Because I've got one. Oh, no, there's one.
I don't have a question, but just to say that if you want to get sort of a closer look and feel of these, try and just get hold of either me, Kasuya's down there, Hura, Sarah, or staff, who's probably also somewhere around that will show you.
Right. Thank you, Ernest.
Question.
Yeah.
Any plans for ion channels that are, for example, temperature or mechanically activated or something like that?
So that varies a little bit from platform to platform. On Q-patch, we can do pressure clamp. We can actually jump the pressure quick enough to make a mechanical influence on the channel. So I would say it's already possible. Whether it's good enough for you, please let us know. We will trim it to the best of our ability.
Okay.
Yeah.
Great. Thank you.
All right. Drinks? Oh, there's a closing remark from somebody?
Yes. I don't know who's...
Drinks, that's the closing remark.